It's a great pleasure inviting you again to this webinar. This is the 74th in the series of webinars that we are conducting on COVID-19. And I'm very glad that we are able to continue this with the help of our friends, both in India as well as from America. And today we have a very distinguished speaker, a very knowledgeable man, particularly in pathology and hematopathology. And he'll be introduced by Dr. Bhagyalakshmi, our professor and head of the Department of Pathology and Vice Principal, Andhra Medical College. And this series will go on and on until we come to the end of this COVID-19. And I would hand over to Dr. Bhagyalakshmi to introduce Dr. Upendra Srinivas, who will be taking us through the early diagnosis of the fungal infections in COVID-19. And over to Dr. Bhagyalakshmi to introduce and start the proceedings.
Thank you, sir. Principal Dr. Sudhakar. Welcome you all to the 74th CME from Andhra Medical College. And first of all, I appreciate all efforts of our Principal Dr. Sudhakar and his time and efforts. And today's topic is apt topic, like it is early diagnosis of fungal infections in COVID-19. So today's speaker is Dr. Upendra Srinivas. And let me introduce Upendra Srinivas. Dr. Upendra Srinivas, he is an alumni of Andhra Medical College. He did his post-graduation MD Pathology from the PGH Chandigarh, did his DM Hematology from AIIMS New Delhi, the only DM in pathology specialties in the south of India. He worked in leadership roles, laboratory directorship in several national and international tertiary care hospitals, and oversees all the laboratory specialties. And Dr. Upendra Srinivas, he is a regular faculty in national and international conferences in pathology and laboratory specialties, and active editorial member in several journals in laboratory specialties. And Dr. Upendra, he is on the executive committee on several pathology associations and has more than 40 publications in national and international journals to his credit. And is currently senior consultant pathologist and hematopathologist and managing director at Quantum Specialty Diagnostics, which he recently established in Visakhapatnam. So, Dr. Upendra Srinivas, over to you.
Yeah, thank you, madam, for that generous introduction. And good morning, ladies and gentlemen. I believe most of the audience is mainly students. And at the outset, let me first thank Principal Sir for giving this wonderful opportunity to share my thoughts on this particular topic. And this is a great platform, as I understood that this has been 74th episode of running this CMEs on COVID as a theme. And I am delighted to see Dr. Budharaju joining us from US. And these kind of platforms usually get us all together and discuss on an issue that troubles us day to day. Let me quickly share my presentation. Let me deconstruct my topic today is to run few slides to explain the context and relevance of this particular topic. And so in other words, it is going to tell us where we stand and what is the burden of the disease that we are dealing with in terms of infections and fungal infections mainly that come along with COVID, and why such an outbreak is happening is another area that I would like to deal with, and how is it diagnosed finally. So if in millennial jargon or millennial lingo, if you want to get to the top of any issue, what we actually do is to understand the gist of it from available studies that are there in the literature. In essence, it is like, what is trending. So if you have to get on top of any issue, as I understand that predominantly the audience is of postgraduates and students, you need to look at the data that is available on that particular current burning issue. So my format sort of is going to take that as generate some discussion points. And I have chosen a few meta-analysis. Meta-analysis is something in general words, you can say that what is trending. It is a compilation of a large amount of studies that were published on a similar topic, like in this case, it is a COVID-19 related fungal infections and other infections, and to see what is actually the trend and what is the gist and distilled information that comes out of a detailed analysis and statistical analysis of these particular studies. So what I have chosen is with the search words like fungal infections in COVID, I've come across 3 meta-analysis, and no surprises that all this data is emerged mainly from the US, China, and Europe, Spanish and Italian articles. And not surprising again, you don't have much data coming up from India for reasons that are better known to us. So in these kind of, I chosen this meta-analysis about 3 of them. The biggest one coming from University of Wisconsin, and where they did a review of almost like 7,000 articles. If you see, that articles are basically systematic reviews and reviews, and they have compiled it into 3 meta-analytical articles. And this is the systemic reviews on about 70. And mind you, the kind of literature that has poured in since the outbreak of this COVID is at the rate of 20 articles per day for the last 1.5 years. So, and you have a minuscule of that generated from India. It's only about 0.1%. So most of this data, actually what it suggests is these infections, a sudden outbreak of infection that we are seeing in the second wave, were there present even in the first wave also, but not to the extent of in epidemic proportions. So this data actually tells us, let us go back to the largest data, is the third one here, which has done about a review of about 7,000 articles. Out of that, they've distilled the gist of 120 articles in which 15,000 patients case studies were studied. And then they have come out with a statistical points like pooled prevalence of these infections that happen with COVID-19 infection. So these are the articles that were put into that meta-analysis articles. And then, so this was the article that I was referring to. In all these meta-analytical articles, there was a concurrence that in most of this published literature, one large such study was happened in China too, and they also concurred that 1 in 5 patients who have developed COVID-19 either have a co-infection or a super-infection. Co-infection is something that it has come along with COVID, or it is already pre-existing in a COVID-19 patient. And a super-infection is something that once a patient gets admitted with a diagnosis of COVID, and this super-infection is something which is developed by the patient while getting admitted in a hospital and getting treated in a hospital. And most of this data, as I told you, is from China, US, and Europe, and it's an adult-based data. It is all retrospective cohort series predominantly, and you have all this patient data developed from ICU as well as non-ICU settings. If you split that, co-infection is something, as I already alluded to, is something which is already there or it just come along with COVID-19. And if you have to split up this, what are the infections that can come along with COVID? It can be bacterial infections, it can be viral infections, it can be fungal infections. So the common bacterial infection that occurred as a co-infection to COVID, these are all data in that meta-analytical article. And this is the common bacterial species that were identified is Klebsiella pneumoniae, Streptococcus pneumoniae, Staph aureus, and these are the common bacteria that co-infected along with COVID-19. Co-infection, the reason why we need to understand this profile of co-infections and super-infections is they are the ones which have determined the outcomes of COVID-19 infection in hospital settings. So co-infection is something which is actually prolonged the hospital stay, and super-infection is directly related to the mortality of COVID-19 associated mortality and morbidity. So what you have seen here is a different species that is there co-infected along with COVID-19, and in a super infection context, it is going to be Acinetobacter and Pseudomonas are the predominant species that actually caused a major mortality in COVID-19 associated infections. So not only bacteria, you have viruses also coming along with COVID-19, and the common viruses is the general seasonal flus, the rhinovirus. These are co-infected along with COVID-19, and the ones which actually not much of burden as far as superinfection with virus is concerned, probably because of competitive inhibition. And surprisingly, in the Western data and the Chinese data, this is all the data which is developed during the early first phase of pandemic. So in this, it is all about Aspergillus and Candida rather than Mucor. And Mucor is something which has come to notice in India very recently, probably because of the environmental conditions or because the kind of conditions that prevail that facilitate molds like Mucormycosis to infect in Indian population. So what we have seen essentially in the Western data is all the white fungus or the Candidal or the Aspergillus. They are the predominant species that actually co-infected and superinfected in COVID-19 patients. So one has to be mindful of not only black fungus has gained importance because of the public awareness, and there's a huge amount of debate in the public discourse every day about this fungus. As we speak, I think it is about over 30,000 mucormycosis cases that are detected in India so far, with a significant amount of mortality. And in addition to mucormycosis, which I'm going to deal in the later slides, we have to keep in mind other possible infections which are already described in the Western literature, like invasive pulmonary aspergillosis. Different terminologies were even given in the media, like based on the color, and there's a debate whether to call them by names or whether to call them by color of the fungus. The color of the fungus, it keeps changing in according to the tissue in which these fungus actually grow. So we have to keep in mind about aspergillosis. This is one such data. There's a new entity that is actually coming up, which is called as COVID-associated invasive pulmonary aspergillosis. These are certain of fungal infections, as we all know. They are they come with a very high mortality rates, and they have to be detected and identified at an early stage so that we can prevent at least a bit of this mortality that comes associated with that. So another entity like candidiasis is again re-emerging entity causing fungemia and leading to the mortality in COVID-19 patients. These are some of the fungus apart from Mucor that has to be kept in mind when we are dealing with patients who are admitted in ICUs and non-ICU setting as well. So one such fungal infection which has come to alarming notice in Indian context is Candida auris. The reason why one needs to be vigilant and able to identify this sort of fungal infection much early is because this fungus is resistant to the conventional antifungals that we use, and so there comes the importance of identifying this particular species. So apart from Candida, invasive pulmonary aspergillosis, and there have been reports of pneumocystis carinii as well, but this is all in Western literature. The reason why we don't generate this kind of data in Indian context is that because we sort of don't test them quite often, and then go by the clinical judgment and in how it actually happens in ICUs and non-ICU settings as well. So unless we use the tools that are at our disposal, it is sometimes all these diseases can be easily missed. So the recent data that we have now coming to the mucormycosis in India, this is a relatively new phenomenon which has gained public importance and public awareness coming up in news media and other media, is black fungus, and with all scary figures and scary clinical pictures, and this has become a very alarming to the general public at large. So few studies, again multi-center epidemiological studies that happened in India, I mean, they are not of significant numbers, but what they have actually concluded in those two articles is that most of it is actually happening because of underlying comorbid conditions like diabetes mellitus and people who are undergoing chemotherapies in oncology related settings, and then people who are undergoing dialysis and all this, and steroid overuse and antibiotic overuse. All thus have been attributed as major causes for mucormycosis in India. And this is one of the, in fact, hardly any studies that have been published in recent. This is as early as in this month. It was published in the literature for all to see. And this is another such review which I gathered just this morning, which again essentially talks about same, but they actually concludes no such kind of, because it's a growing. When was the last time that India or any country for that matter has seen a fungus growing in epidemic proportions like this? So these kind of epidemics, when they break out, the dictum is that test, track, and treat. And quite often we fail in testing and tracking, and then the treatments are largely based on empirical ways. So the second topic that has to come into this picture is, why is this happening? A lot of theories have come both in the scientific literatures, as we have seen in earlier articles, and even in the public discourse and media debates and arguments, where they have finally attributed few reasons why such things like proneness to this kind of fungal infection in India is happening, starting from uncontrolled diabetes and steroid abuse, antibiotic abuse, and when the oxygen supplies were in short supply, they have to be prepared in such a short notice, and the way they were delivered in unsterile containers to the hospitals. All those were the hypotheses that were put forward in explaining the sudden outbreak of these fungal infections in India. So, and there was a recent report that probably this must be coming from the nasal swabs that we use in doing COVID RT-PCR testing. So these are all hypotheses, and no concrete studies that can pinpoint to these factors have been established and published in the literature. And one strong, if you look at one strong point theoretically in all the above listed things, is that generally there will be an immune weakness. The immune system is the one which is predominantly attacked by this virus, other than the lung. The lung is an entry point, and predominantly what is affecting is the immunity. And genetic, probably some of the genes that are involved in developing the immunity are also attributed as causes for why these kind of epidemics are breaking out post-COVID in countries like India. So before we jump into understanding the immunological mechanisms that are predisposing patients to fungal infections, the basic parameter, the hematological parameters are the very common ones that we actually look at when the patient gets admitted. This sort of provides us an opportunity as a window to see what exactly is happening to the blood post-COVID infection. So there's a little unique features as far as this virus is concerned. And usually the first cell response of defense to any virus is lymphocytosis. In this scenario, in the context of COVID-19, what happens is the lymphocytes are the ones which are first destroyed. And this is much akin to what we see in HIV and other influenza virus and hepatitis virus. Usually the response is lymphocytosis. What we have here is a lymphopenia. And the other unique feature is a distinct feature is usually in neutropenics. When there is a neutrophil is the first line of defense for fungal infections. In neutropenics, when the neutrophil count is down, those are the people which we often come across in hematological problems. The neutropenics are the ones which are likely to develop all sorts of fungal opportunistic infections. But in this setting, in COVID, we have used a parameter like increased neutrophils and decreased lymphocytes as a harbinger of a very severe COVID disease. So again, with a large amount of neutrophils available and a lymphopenia predisposing to a fungal infection and other opportunistic infections is something unique in COVID-19 infection. Probably because even in abundance of neutrophils, such thing is happening, it means that there is a problem in the innate immunity, which I'll come to in the next few slides. So that makes the neutrophils less functional and they're not able to do their job. So some of the other parameters which are actually gaining importance day by day are, you see basopenia, and you will see decreased in natural killer cells. And then these are the basic parameters are not, these are some of the parameters, the specialized parameters are not detected by the machines that we have in routine laboratory. So you need to do a bit of flow cytometry to identify the proportion and the function of NK cells. And just like some other viruses which are directly attacking platelets, COVID-19 is also seems to be affecting platelets, causing not only thrombocytopenia, but also predisposing to thrombotic conditions. And a few of the studies coming from the Europe has identified a certain subset of eosinophils are increasing in COVID-19 infection, which are CD62L subtype of eosinophils, which again can be detected by flow cytometry. That are the patients who are most likely to get into an ARDS and a severe type of COVID-19 disease. So if one is interested, this is an article which sort of discusses about length and breadth of what happens with the hematological parameters in COVID-19. So coming back to the immune response and why such immune weakness is happening in COVID-19, thereby predisposing to a whole lot of opportunistic infections. We're just seeing only fungus. It may be something else that may come up tomorrow. So immune responses basically are divided into innate immune response, which is actually contributed by the cells like neutrophils, monocyte, macrophage lineage, and tissue macrophages, the Kupffer cells, and all that, the histiocytic macrophage activation system cells that contribute the innate immunity. And the other arm of immune response is usually an adaptive response. Adaptive means it's a trained immunity. These cells get exposed to antigens and then they trigger a whole sort of other immune regulator downstream and thereby contribute to the trained immunity. So, and by and large, if you look at the some of the studies which were trying to establish why the immune weakness is happening in COVID-19 patients, most of them seems to be affecting the innate immune arm rather than the adaptive immune response. Adaptive immune response, by and large, is the target for HIV. So in innate immune response, if it is attacked, what is going to happen is something which we'll see in the next few slides that explains the pathophysiology behind COVID-19. So this is one such article where I have found very, very interesting findings. This has come up in a journal called Cell, which has got a very, very high impact factor. This kind of research we hardly get to see in Indian context. And this sort of tells us exactly at cellular level what is happening. I mean, once you read this article, you will be tempted to believe a lot of conspiracy theories that are actually, I tend to believe that this conspiracy is no more a conspiracy, that it is a lab leaked virus. This is a virus which is designed in the lab by picking up properties from different viruses. This is possible in the present day technology like gene editing that happens with CRISPR molecules. And then this was stitched in such a way that these develop different properties like transmissibility, and this is all come to under one category called gain of function, which is a term which is actually coming more and more into public. And gain of function is something you are making a virus more virulent in terms of immunogenicity, transmissibility, and then you by literally passaging them into multiple cultures and make it gain some more functions in terms of mutation, in terms of developing capability to mutate itself and all that. Once you read this article, it sort of tells us what exactly is happening. So this virus is taking, though it is taking an entry into the lung, what actually it is doing to the maximum damage is to the immunological system. So this is a schematic diagram which explains what exactly is done in this particular article. Right from the point of entry of this virus by adhering with the spike proteins on binding to the ACE2 receptor protein and then getting internalized, and then activating the cytokine storm and everything, all that studied with different tools, and they have come out with very interesting points. So if this is what happens in cytokine storm, it has to be depicted in a picture. This is how it looks. Whole lot of interleukins, and these are all nothing but a chemical substances that are released by the cells of immunity, predominantly of those innate immune cells. And this is what happens with their circulating levels as one moves from asymptomatic stage to the final critical stage of developing ARDS and subsequent mortality. This is how the cytokine storm picture looks pictorially. So, as I said, the innate arm, the cells like neutrophils, the dendritic cells, and the macrophages, all these actually contribute to the significant amount of this cytokine storm. And this is what happens in different phases of COVID infection, from mild to moderate to severe, and different cells were put into the milieu where they develop all these molecules that contribute the pathophysiology of COVID infection. So, coming to the genetic is a very interesting point. So this is basically two studies actually concurred on the same observation, that there is some problem and mutations happening in the chromosome 3, where a gene for dictating how an innate immunity of an individual is going to behave. So probably these are some of the interesting findings that can find a way to from bench to bedside very quickly. And apart from that, this virus also, as I told in earlier slides, is going to attack the platelets, not only decreasing their number, but what happens is once it attacks a platelet, it makes the platelet to release all its procoagulant substances, and thereby producing a clinical picture like a MAHA, microangiopathic hemolytic anemia, if not a DAC kind of a picture. DAC sets in when there is a co-infection with a bacterial or a fungal infection. So it is going to affect the blood and the coagulation system as well. So apart from lung as an entry point, and then you have the immune system which has gone berserk, and then you have a blood system which is causing a significant mortality in terms of precipitating in a MAHA kind of a picture and a DAC kind of a picture. So coming to how do we diagnose these fungal infections? Traditionally and conventionally, we do them with fungal cultures. The only disadvantage of fungal cultures is that they take time. And if you have to have a fair degree of suspicion of a particular fungus is the cause for infection, then it takes almost 5 to 7 days. And if you need to know the susceptibility also, add another couple of days for that, and it's going to take time. So some of these tests that I'm actually going to describe, which has come to the surface, are the ones which are already existing from the past decade. It's only just that we have seen this epidemic. All these tests are surfacing again. So the advantage of non-culture tests is that they can diagnose even before the culture shows up. So we can detect the earlier signs of infection with the fungus in the blood by different modalities, and identifying different constituents of this fungus. You can either identify them as an antigen, which can be a component of the cell wall of the fungus, like mannans and galactomannans, or you can see whether already the patient who is infected with fungus, they have developed an antibody to it, or as I told, Mannan tests are the ones. Mannan are molecular moieties which are present on the cell surface of the fungi. And there are certain other glycoprotein and associated tests which has come again of some importance in screening and identifying those high-risk individuals who are likely to develop these fungal infections. These are some of the important tools that can identify and early detect some of the high-risk individuals. And molecular tests also have started coming up, and PCR-based, PCR-based tests, they have the advantage of not only detecting the particular species of the fungus, but they have ability to detect certain of these genes that are responsible for resistance to particular antifungals or not. So all this is possible in the with the current day technologies. So this sort of article describes, this is as recent as 2020, and this tells us where we stand and what is the landscape of fungal infections and what is the approach in diagnosing them. So these are the list of tests that we have in current day. As I told, ranging from 1,3-beta-D glucan. Glucan is basically a polysaccharide moiety which is present on the cellular surface of the fungus, and different modalities have different sensitivities and specificities. And some of these tests really enjoy a very good sensitivities and specificities, and some of them have a very good negative predictive value. If they are absent, the infection is absent for sure. So these are some of the tools that largely are used in the Western context, but hardly we get to use them in Indian because of more because of non-availability and probably some part because of lack of awareness. So this sort of presentation is to bring up these points so that these can be used in the right settings, and then probably able to detect more of such fungal infections in our COVID-19 patients. So these are the list of tests that we have, and this was in the context of candidal infections. And candida, in fact, is the commonest fungal infection. And aspergillus, we have a different set of diagnostic tools ranging from serological testing to molecular testing. So the strategies, this is a review article which talks about what are the strategies, why do we have to do a molecular study, because the molecular studies by and large have proven to detect early, to make an early diagnosis of fungal infection to the extent that it detects much before a clinical manifestation of a fungal infection or even a radiological observation of a fungal infection. Almost a week's time before you actually see them clinically, or at least 3 to 4 days before you actually detect them radiologically. So this is a proven literature where the molecular studies have been used and found to have made a significant impact of identifying these fungal infections and offering better treatments and averting the mortality that comes with it. So this again is an article, interesting article, which comes up with a lot of recommendations for different clinical scenarios and clinical contexts where all these tests have shown a significant clinical value, and they contributed to reducing the mortality associated with fungal infections. And so these are all similar, different scenarios in both in ICU settings as well as in non-ICU settings as well. They've shown, these are all the recommendations that I've put forward in this very interesting article. Coming to the tests that are available now in a very few places in India, we do it here. And 1,3-BD glucon test is again a chromogenic based assay, which takes about less than about 8 hours. So that there comes the significance and the importance of this particular test in identifying individuals who are at risk, and then treating them with the appropriate antifungal and mitigating the mortality. So this is again a review. The conclusion of this review was that this test enjoys a good sensitivities and specificities and has a very high negative predictive value. And this assay also in not only in detecting and identifying individuals who are having an early signs of fungemia, but these tests can be used in individuals as a tool for monitoring antifungal therapy. This sort of shows us the numbers about their specificities and sensitivities and the percentage of negative predictive value. And they have come forward with few recommendations in what context and what scenarios you can use this investigative tool. And there are a lot of other fungal detection modalities, which is like lateral flow assay, which again is a point of care testing. Point of care testing, ease of doing it and quick turnaround time is going to help us in detecting these infections very fast in ICU settings. And all these these kind of lateral flow assays are available for detection of Candida, Aspergillus, for that matter, even in Cryptococcus. Cryptococcus may not be relevant in COVID context, but Aspergillus, Candida, you have this quick test, and we can, with this kind of instrumentation, we can actually densitometrically evaluate the quantity of that kind of antigenemia. So this is another one specifically required in identifying the antigen specific to Aspergillus is galactomannan. So this test is not new; it is already there, available and used in different clinical contexts, and we can use this galactomannan assay in detecting individuals who are at risk for Aspergillus. And more such products are available in detecting specific antibodies to the fungi, Aspergillus and Candida, and even in Cryptococcus. So with this, the essential point that I actually want to drive in here is tools are available unless we test them, unless we liberally use these diagnostic modalities. We tend to often, miss these kind of infections and will not be able to come up with the correct numbers of prevalence and incidence of these kind of infections that come along associated with COVID-19. And co-infections and super infections is a clear and present danger. They're going to come along with COVID-19. And this virus is very unique in many number of ways, as I've already alluded to, attacking the immune system. We don't know and we can't even predict what is going to happen in terms of profile of opportunistic infections that we can come across in future. So one has to be really vigilant and updated with the literature as to what is happening as far as this opportunistic infections happening in the background of immune weakness in COVID-19 patients. With that, I rest my case, and then any questions are welcome. Thank you, madam.
Thank you, Dr. Upendra Srinivas. So this is an excellent presentation. You have touched upon all meta-analysis, data, statistical analysis, and immunological parameters and hematological parameters and immunity in detail. You have described very good. And molecular studies which are useful and by which you can early diagnose the fungal infections, which is most important currently. And like what you have suggested is 133 PD glucan test, which is a chromogenic method, which is most important. We can give the report as early as possible. So good presentation. And as far as our King George Hospital is concerned, so we have been sending regular reports to the government. And in Visakhapatnam, we are having almost 8 to 9 hospitals where we have been doing the mucormycosis treatment. And only in KGH, so far, we have detected case reports of mucormycosis in post-COVID-19 is 167. And total deaths among these is almost around 16 total deaths for the last more than 20-25 days only. And as far as we are concerned, what we have been doing for all these cases is we have a very good teamwork by anesthetists, neurosurgeons, and our principal, Dr. Sudhakar, ENT surgeons, and ophthalmologists. So all of you, with the teamwork, we have been doing the very good study. And like we have been doing the frozen section of the specimens, as well as the biopsy of the specimens also. So far, we have received 86 cases of from the neurosurgery department. And within this 86, almost 84 cases, they have showed the mucormycosis. And 2 cases we came across combined aspergillus and the mucormycosis. So predominantly in all cases, we could see only mucormycosis and associated fungal infection is aspergillus niger, which we have observed. And we have been studying the 2 studies we have been studying. One is comparison of the frozen section along with the histopathological biopsy correlation. That is one study. And second one is clinicopathological study of mucormycosis in post-COVID patients. So far regarding our data, so what we have observed is within the both frozen section as well as the biopsies is very few cases, almost all the cases, they have showed extensive necrosis. So whatever the material we have received, it showed extensive necrosis and the karyorrhexis, which is the breakdown of the nuclei, along with that the granulation tissue, as all of you know, that is because of the infection, and the thrombosed blood vessels. Most of the cases, what we have observed is number of thrombosed blood vessels are seen, and no doubt we have seen the fungal angio invasion. And some of the cases, extension of the eyeball has done. In these cases, we have observed the perineural invasion by this fungus. And apart from that, other findings are dense lymphohistiocytic proliferation, foreign body giant cell reaction, and extensive areas of hyalinization. So these are our studies, and we have been doing the frozen section as well as the histopathological study. And what we have been doing is we are trying to give the report as early as possible within 5 days after we get the biopsy. So no doubt in the frozen, we have been giving within 15 to 20 minutes or maximum by 30-40 minutes. And biopsy also we have been doing, giving the report after 5 days. And moreover, the diagnosis of these fungal infections, everything depend not only on the diagnostic work, you need to correlate with that of the clinical findings, radiological findings, and all these, it's a multidisciplinary approach. We have to do it with the final diagnosis, whatever it may be. The early diagnosis is most important to prevent the mortality and morbidity to the patient. So thank you very much. And now we invite comments from Dr. Chilsunis, Dr. Sridhar and Buddharaj sir are in online. Dr. Sridhar, comments from Dr. Sridhar.
Before someone pitches in, ma'am, I'm pleasantly surprised with the number of cases that you have described and the kind of findings you have come up. I think it's time we can contribute that kind of literature to the ones which are already existing, and that will enhance the kind of robustness in the statistics that we have. India hardly I mean, at the same time, I'm even very scared that if in KGH alone, if we have so many cases, imagine about countrywide, and probably the figure of 30,000 may be less. And that sort of tells me that what is the magnitude of this problem is going to be in the coming days in India. So, I mean, once we use the screening tools that I've described in my presentation, I'm sure it will be more prevalent and are likely to have more bigger numbers as far as this mucormycosis is concerned. So that's a very impressive data coming up from KGH, ma'am. I think it's time that we can contribute this to the literature, existing literature.
Sure, definitely we'll do that. Our principal is after that only has been inspiration to all of us. So have been doing that work.
So I think Dr. Chilamuri is waiting for his microphone to be unmuted, I think.
Yeah,
Meanwhile, Dr. Buddharaj said, okay.
No, so Dr. Bhagyalakshmi, I think what you're doing is what we do. I think I liked the earlier presentation of all the tests that are available, but they're not practical even in the United States. The way you're going about diagnosing and giving immediate feedback to the surgeon is the way we go about it. The only way to manage mucormycosis is to have a high index of suspicion and do what you exactly do it. Get a specimen, look at it, and give feedback immediately to the surgeons to go further. All the other tests are great, but you really don't have time to get any of them. And in the United States, we don't do any of them. We do an immediate gram stain or a KOH, just like you did, and proceed with treatment based on that debridement. And of course, for us, amphotericin B is relatively easy to get. And we're trying our best to get it to India, but the custom services are giving us hell in importing these drugs to India. But hopefully, we'll get over that. But I think the earlier presentation is excellent. But I think we need to look at practical solutions. One other question I have is we still are not sure all the risk factors that's causing this excessive mucormycosis. We know about the use of steroids, but we've also seen people who have not used that much of steroids getting mucor. So that's intriguing. And the second thing is this steaming thing that people use often at that high humidity, is that increasing the chances of getting mucor? And the presence of DKA and diabetes always is risk factor. So it's unclear in India why you have such excess of mucor. Then everybody describes the disease, but they're not pinning down the risk factors. What were the interceding risk factors that cause excess? And we are getting worried about whether the Delta variant suppresses immune system more than other viruses. There's more recent basic science research suggesting that as the virus is mutating, it's becoming more virulent and targeting T cells more than the previous variants. which is exactly what influenza did as it mutates every year. So we're getting worried whether the Delta variant uniquely makes us susceptible to mucor. I think that's what we're looking for from your observation, if we're seeing that in India. That's the comment I have.
Thank you, sir.
I had a couple of questions for Upendra. So we know that aspergillus grows pretty quickly in cultures. Usually within 2-3 days you can actually grow them. Probably not so with mucor. We have an advantage of galactomannan and 2D beta glucan for aspergillus. We don't have similar tests for mucor. So the second question I had was, do we know what is the predominant species of mucor currently in Andhra, at least, if not in India? I know that PGI Chandigarh is collecting large volumes of data in terms of the species as well as host factors, and trying to come up with some answers as to why we're seeing so much of mucor. Yeah, true. Like Dr. Chilamuri said, we still depend on methenamine silver stains and things like that to identify mucor or aspergillus. You hear some of very rare big institutions, some of us who see a lot of aspergillus and mucor in the transplant setting. We tend to have some availability of lateral flow devices, but I don't think there's any use for lateral flow devices for mucor. That's mostly for aspergillus again. I haven't heard at least. And I'm hearing that the lateral flow devices actually have pretty equivalence in terms of, predicting with, PCR in cultures, and probably a very quick way to identify the species. So if Upendra or Dr. Bhagyalakshmi could, tell us if they're seeing a particular species of mucor in Andhra, or is it, multiple species?
The first part of the question, I'll take it. In the second part, I'll leave to Dr. Bhagyalakshmi. So the first part, as you said, in the context of mucormycosis, the rapid detection only comes to the molecular method. And there are no other serological methods that are available in detecting mucormycosis. One such as a frozen section or a KOH mount, and fairly sort of tells us that we are dealing with mucor. There's no problem with that. But by and large, the rapid molecular tests that are capable of identifying the subspecies of mucor also. These kind of tests are also now available. Most of it is actually coming from China. So most of the kits that we are using actually are coming from China. And yes, for the context of mucor, molecular tests are there, and which actually can detect the species and subspecies of different types of mucor. And I don't have enough data to suggest which kind of species is more common, or I think that part of the question I'll leave it to Professor Bhagyalakshmi. Madam.
Yeah. So what we have been doing is we have been doing the special strains and silver implication in biopsy. And simultaneously, our microbiology people are doing the KOH as well as the culture also. The species and everything, it will be, you can easily diagnose by the culture report only. So once we correlate with that of the microbiology reports, then we can easily tell what type of species is predominant in mucormycosis. So that we have been doing that workout, and we have to get the statistics from our microbiology faculty also, then we can tell.
So the frozen sections in KGH right now, so the OTs have the availability of frozen sections for all mucor surgeries, and you're able to give them an answer as to if you're seeing a HiFi and frozen section, or?
Yeah. So what we have been doing is, so we informed all the residents and house surgeons whenever any surgery is there, they need to keep the specimens in 2 samples, like one is in the saline and one is in the 10% formalin. It is meant for the biopsy. So from the saline sample, we have been doing the frozen section, and within that frozen section also, we have been doing the PAS strain for the clear-cut identification of the mucormycosis. So it's very, so by practice, we came to know that we can easily diagnose in the frozen section also. And we have been giving the report, and a final report usually after 5 days, correlating with that of the biopsy, the final report we have been giving.
That's truly amazing that you're doing it. This is what India does well than any other country, and I'm very proud of what you're doing. That is stunning, because we struggle with that even in the United States. But I think that's your experience and seeing so many. That's what the experience gives you, and it's amazing what you're doing. Maybe you should work with us, because we struggle with this with our pathology and microbiology. That's stunning what you're doing. I congratulate you.
Thank you. Thank you.
Is mucormycosis really as big as a problem in US as it is in India?
No, it's
So-
not. We saw very little when COVID was at its peak. We hardly see any COVID now, but we saw more aspergillosis. So they were underdiagnosed in the first wave. So we didn't do enough testing to look for aspergillosis. So we missed it. In the second wave, as we got more aware of it and became more aggressive, I think nearly 20% of critically ill ICU patients may have aspergillosis before they died. So much more common to see aspergillosis than. So in our experience, we just saw one mucormycosis with COVID. And that we accidentally picked up as we weren't aware this was creeping up on us. So we kind of were a little delayed in picking it up, but we picked it up. He's alive, but lost his one eye in the process.
Sometimes it's difficult also to identify the mucormycosis. With experienced eyes only, like, yes, in this necrotic material, definitely there should be mucormycosis. So we have been sparing a lot of time to find the hyphae and fragment it, sometimes because of the processing and everything. We may not able to get the intact hyphae and everything. So most of the times, 50%, it's very easy to diagnose. Whereas in some cases, we need to do the complete search for 10 minutes, 15, 20 minutes like that to find the fragmented fungal hyphae. And for that, the special strain is counting a lot. We have been reporting by doing the special strain, particularly PAS strain. We have been doing regularly for all the cases for the frozen as well as for the histopathology sections.
Yeah.
Yeah, for every 25 or 30 pulmonary aspergillosis in the transplant setting, in BMT setting and solid organ transplant setting, we probably see one mucor for every 30 or 40 aspergillus. So pulmonary aspergillosis is what we see mostly. I mean, mucor, I haven't seen any. We treated probably close to 2,000, 3,000 COVID infections in our hospitals. And I haven't heard of any mucor infections, but we did see a few pulmonary aspergillosis in immunocompromised individuals who had had COVID infection as well.
Okay, sir.
So what is the Sudhakar, what is the availability of antifungals now? Is amphotericin B now available, or are we doing posaconazole, or what are we using?
Yeah. Sridhar, thank you very much for those wonderful comments. And the present status is that we have an improved supply of the amphotericin B, but not to the extent that we need it. Like we're getting it for about 20% or 20 to 25% of the patients. And for another 50% of the patients, we're getting the posaconazole injections. And the rest who are in the early stage of the disease and all, we are giving them the posaconazole tablets. And we're giving the posaconazole tablet for those people who are on the follow-up as well. Now that this is almost 3 weeks, some of the patients are discharged and they're coming back for the review, and we're giving them the posaconazole tablets when they are in the home treatment. So that's where we stand now. But I think still it needs to improve. Recently, we have made the drug control authorities to give permission to the Reddy's laboratories to manufacture this amphotericin B. They already have a license to manufacture that in Hyderabad. but there is another plant nearer to Vizag in Paidippuram in Srikakulam district, where they have a plant which is capable of manufacturing this amphotericin B. And we have coaxed them to give permission, and they have given the permission. I think it would take about around 2 weeks for them to get all the validation done, it appears. And so once that happens, perhaps we will have a larger quantity available for the local use.
We approached the Indian embassy to help us clear shipments of amphotericin B from the US. So the Vice Consul is dealing with it. At least in another embassy in Georgia, they were able to help expedite shipments to, I think, Hyderabad or someplace. So we're trying for Visakhapatnam, but struggling with UPS and the government of India to clear them. But they're insisting on asking us for patient names, which we said this is going to an institution which will administer to whatever patients they have. There's some, they want us to send through Prime Minister's fund or something, but that doesn't reach Vizag. It'll go anywhere in the country, and we raised it to go to Visakhapatnam. That is our goal. So the Indian embassy is still working on it. I wish there was less politics in delivering important medication, but I guess we have to live with what it is.
So, Principal Sir, can I join, sir? Principal Sir.
Yeah. Please.
Sir, good morning, sir. Good morning, sir.
Yeah.
Thank you, moderator. Wonderful talk, Upendra. This is to Sridhar, sir. Sridhar Chilburi, sir. Sir, you said customs issues, no, sir. Sir, we are in touch with the local customs in charges, sir, who is in charge of clearing this any international shipments, COVID relief shipments, sir. We spoke to them, sir. They said they will clear it on a war footing, sir. Not on a priority basis also. They said they will clear it on a war footings. They have guidelines, they have instructions, clear instruction from central government, customs authorities and everyone to clear all COVID relief charities or whatever philanthropies on a war footing, sir. The person superintendent who took care of this cryogenic cylinders, all those imported cylinders recently, no, sir. I was in touch, sir. Any product shipment to AMC or KGH, directly they should land in Vizag Airport or Seaport, sir. He said he will clear it in express basis, sir. Mr. Raju, sir, he said he cleared all those cryogenic cylinders and all those things.
Yeah.
Okay,
I think
so I'll ask them if UPS will shift it directly to AMC Vizag. Then we are told everything goes through Hyderabad, and that's where the bottleneck.
Yes, sir. They said any other seaport or airport, we can't help, sir. But if it is directly to Vizag Airport or seaport, they said they'll clear within no time. And we have all the waivers now, sir, from Finance Minister, Union Finance Minister regarding GSTs, all those import duties for this COVID relief material. Until September, we have, sir. As of now, they have waivers, sir, for import duties and all those things from Union Health and Finance Minister, sir, regarding COVID relief materials, sir. GST also is tremendously reduced from 12-18% to 5% for all COVID relief materials.
Yeah, Naveen,
yes, sir. Yes,
Naveen,
yes,
we
sir.
have to ensure whether the policy is same for the drugs as well as the other material. Now that you have the oxygen concentrators and few other material
yes,
that will be coming from abroad,
yes,
we'll have to find out whether the policy is the same for the drugs also, because sometimes they'll be very critical with the drugs. So we have to be careful with that. And if that is so, if you can find out if that is the same, it's okay. That is number one. Number two is what about the place from where it is coming to this particular place? It will be all right the moment it lands in Vizag. But what are the regulations that are prevailing in a place where they have to be shipped from, like in the United States? From which state would they really take it on board to be transported to Visakhapatnam? These are the two aspects I think we have to have an eye on that.
Yes,
Yeah.
sir. I'll connect him to you, sir. We'll clarify all doubts and put across to Dr. Sridhar, sir. I was specifically
Okay.
asking for amphotericin B only, sir, actually. That was the
Yeah.
crux of the problem presently. Like you said, position just improved, sir. Just slightly improved.
No,
Slightly improved.
we want to help. This is we have many people who are going to help us get the medication. I've talked to pharmaceutical companies. So we have doses that can be shipped fairly quickly. So let me ask them UPS if they'll ship to AMC.
So,
Yes.
Sridhar, have you talked to General Gada Sudhakar, API president, or?
No, I did.
So they
No, I
were
haven't.
set. So they had sent a large numbers of oxygen concentrators through API. I talked to Sudhakar, and I think we might be able to talk to API guys, and because they have the
No,
transportation
they did
available.
oxygen, but drugs is a different process. That's the problem I had with. So I
maybe
know API
Sudhakar,
sent
API might have.
oxygen. Yeah, did they send amphotericin B? At the best of
I
my
don't.
knowledge, they have.
So I don't think so, but I think they might be working on it. So they might be having access. They are working through Seva International, and
Yeah.
they're using the Seva International flights to transport them. But as far as collecting at the Vizag customs, that should be easy. The collector of Vizag should help us deal with that pretty easily, actually.
Yeah. Okay. Because Seva sends it through Hyderabad. They don't ship directly to Vizag. That's the problem with Seva. So whatever you ship to Hyderabad will stay there. And we really want King George Hospital to get it. That's all. But anyway, I'll try again.
Yeah. And
Thank you, sir.
Dr. Vijay Shekhar wants to share his screen. Let's allow him to share the screen and let us see
Yeah.
what he has.
Sure.
Vijay Shekhar, go ahead and share your content. Vijay Shekhar, are you there?
Yes, sir, I am here.
Yeah. Share your screen.
Sir, are you able to see, sir?
No.
No, sir?
No, not
No.
yet.
Just trying, sir, 1 minute. Okay, anyway, sir, just I'm trying to share. Meanwhile, I would like to mention about one interesting case which we have seen. Actually, one young
Yeah, we
individual.
can see now.
Are you
We
able
can
to
see your screen. Yeah, we can see your.
Yes, sir. So this is one of the case, almost a 50-year-old, 47, 50-year-old male patient presented with that black bleb of the near the elbow region. It has progressed rapidly and to a black spot and a sharp. And unfortunately, because he was amputated, so there is no other go. Above elbow amputation was performed. But still it is, even the stump is, the necrosis is extending and still the culture from the stump also is showing. So we have, but we are planning a disarticulation, emergency disarticulation, but the patient general condition is not good. So I just wanted to know the experience of any of the speakers or senior consultants here. What are the cutaneous lesions? How many cutaneous lesions they have seen? These are some of the interesting cases which we are getting in our ward. This is one cutaneous case. The others are two lung cases also, pulmonary mucor suspected and radiologically also we have seen. So just wanted to know the feedback from others.
Yeah, madam, how frequently do we get the cutaneous mucor? I mean, so it said that it is very rare, but how rare is it?
It's rare only. So far, we have not received any cases of cutaneous mucormycosis.
Yeah.
We haven't encountered.
So in terms of seeing these lesions all over, so when mucor first began to appear in India, we were beginning to worry. whether there was contaminated intravenous liquids or steroids. Steroids often grow if a poorly manufactured fungal infections are contaminants are fairly common in steroid preparations. So at that time, we were thinking this may be one of the source of infections in India. And if that were to be true, we would have seen metastatic diseases like mucor in the blood, mucor in the skin, mucor in peripheral areas other than the rhino orbital mucor. And this case that we are seeing now, that makes me worried how he would have fungal infection so far away from the either he had bacteria, fungal seeding into the bloodstream with spread, or because of invasion of blood vessels, or he actually had some contaminated IV fluids or something that led him to develop those metastatic lesions. That worries me in these cases where you're getting mucor outside of the eye and the nose. That's what I would concern about. The second thing is I would like to see if he has neutropenia or complete suppression of white cell counts when he developed this. Maybe in unique patients like this, granulation stimulation factors may have a value, increases white cell count a little bit. At least this type of mucor, there may be a value. The local one, I don't think colony stimulating factors have a role. But I think when you see metastatic disease like this, we should look at contamination, and number two, what's the white cell count, and if we can bring it up a little.
Thank you, Dr. Sridhar. Sir, one more question, rather doubt I have. We have so far operated almost 7 cases of intracranial fungal abscesses with contiguous spread from the nose and also some from the orbit. So I just wanted to, we could remove the capsule in one case along with the capsule, but others, because we have approached through endoscopic route only, we could drain all the fungal abscess. Will there be any role of topical amphotericin? After the cavity, we scoop out the cavity, take out the wall of the cavity intraoperatively. Can we try? Is there anything I searched but I couldn't find out? Any experience in the audience?
In the literature that I reviewed, I could not find local amphotericin to be of any value. I think with the intravenous, you get adequate tissue penetration. So there's no added advantage of trying local amphotericin B. It was never formulated to work locally, so I doubt anyone is any longer using local amphotericin B. That's what I found.
So the LV Prasad Institute, I mean, across the country, is creating a staging system for rhino orbital cerebral mucor, and it's an interesting staging system. It might actually come out soon. And talking to some of the people at LV Prasad, they have a protocol where they are irrigating the debrided area at the time of surgery or postoperatively with amphotericin. I think Dr. Sudhakar might have some experience with that.
Yeah, no, it's, Dr. Buddharaju, thank you. And I think the experience is the same, like everybody would say there is no harm in doing that, but there is no evidence to say that it will improve the situation in the neighborhood of after you basically debride it. There's no evidence that these people have done better, they have fared better than the others who have not received a similar treatment for the local installation or local injection, either injecting into the periphery, into the normal appearing tissues, or in packing the nose or the paranasal sinuses with this particular impregnated gauze. So there is no evidence as to whether it really works there. That's a problem. The second thing is here, Dr. Vijay Shekhar said that he had removed the capsule in one of the cases, but in the rest of the cases he could not. I'm sure that he had sent that capsule for the histology so that we will understand whether it contains the fungus, in which may throw some light towards whether we have to remove the capsule also, otherwise what happened to that particular patient and the other patients also when you have aspirated, when you have just drained the abscess. So these are the 2 points that need to be understood. I wish that you send the capsule
sir.
also separately. Yeah.
Yes, sir.
And
It was sent
see,
and the pathology, madam, I have seen it, sir. They have reported also the capsule is showing positive for the fungal elements, sir. So it is already proved.
yeah, in that case, it appears to us that from now onwards we have to be very careful, and we should be removing the capsule also. Simple drainage of brain abscess is of very limited
Of
value.
course, sir. Yes, sir.
Yeah,
Yes.
yeah. I think that will give us a cue as to what needs to be done in future.
Yes, sir. But in most of the other cases, so these are contiguous spread almost from the like epidural granuloma or abscess initially at the basifrontal region, at the adjacent to frontal sinus. And so we have no other go because anyhow there is an access there by the ENT surgeons and our neurosurgical team. So that is with least morbidity. Otherwise, what we have to do is we have to do a regular craniotomy, transgress the normal cortex. So it is all, we are also thinking and discussing about the case in our department, sir, regarding each and every case. We think we can do much better in future cases.
Yeah,
Thank
good,
you,
good, good,
Thank
good.
you, sir.
And you guys, Vijay, you have to write this up because you're teaching the rest of the world how to manage mucor, because nobody in the civilized world will have the kind of experience that you have. So you need to write it up, and we're glad to help you write it up because we're really learning from you. You're not teaching us, we're learning from you how to manage this.
So, Dr. Sudhakar, are there like standardized protocols now, either through ICMR or the surgical societies in India? So I was hearing from LV Prasad mostly that they have some kind of a standardized protocol where they're irrigating everybody with regular amphotericin, not liposomal, to the surgical areas.
Yeah, Dr. Buddharaj, we had those 2 speakers, the oculoplastic surgeons from the LV Prasad on our forum as well, here in the AP webinars. In fact, I moderated those sessions, and they said the same thing. But only thing is there is not enough evidence to show the usefulness of that particular irrigation. The same question was raised there also. One is Dr. Ocean Bansal from Visakhapatnam. She is in Visakhapatnam branch. The other one is Tarjini Dave from Hyderabad. So I am aware of the quantification or the staging of the disease that they are doing. But to me, it appears that, it is very gross, and it may not throw adequate light on the management protocol that we need to follow. Actually, I've gone through that during their presentations. However, let us see how it works, and perhaps you also can see where our cases fit in the classification that they're suggesting.
So Dr. M.E. Apparao, sir. Sir, you're welcome.
Yeah, are there any comments from him?
Yeah, he has raised his hand. Dr. M.E. Apparao.
Yeah. Shiva, unmute him.
Not
Yes, sir. Unmuted him, sir.
Yeah. Dr. Apparao, please go ahead and speak up.
I think he has not unmuted.
Unmute yourself.
Apparao, unmute cheyandi. Apparao garu.
Yeah, is it okay now?
Yeah. Please go ahead.
Yeah. As an ENT surgeon, we see cases of ethmoidal polypi on both sides, which are very refractory to treatment. And they are secondary to allergic fungal sinusitis, which is due to non-invasive fungi. We call it as non-invasive fungi. We have made a study also, And all these fungi, aspergillosis and mucor, and all these things were reported in our study. It's all non-invasive fungi, but they are all very resistant to treatment, very refractory to treatment. They recur again and again. We have to operate again and again, even though with the modern armamentarium, the results are far better now. Now, we have seen a few cases of mucormycosis also while I was in office, and we did radical surgeries. Say, for instance, if there is a fungus on the palate and giving rise to necrosis, we did a total maxillectomy and be done with it. We have done a few cases of orbital exenteration also, but when the fungus has extended up to the orbit, what I noticed is the infection now readily extends into intracranium and we lost the patient. And my observation is in the so far why there is such a rampant increase of this particular invasive fungus, that is mucormycosis. Aspergillus, I don't think it will produce so much of damage. But the slide which is shown by Dr. Vijayashekhar is very frightening. So we have to, what I would like to impress upon you is, rather than drugs or surgery, we have to definitely hit the root cause of this particular problem. Why it is happening? It was not so rampant in those days. Update it has become rampant. Even in the first wave, it was not there. Now it is there in the second wave. We don't know what happens in future. Number one, regarding the regarding the amphotericin B, we have treated all our cases without amphotericin B. They survived in the previous setting. And I have some experience with amphotericin B because of the refractory nature of the previous disease, that is non-invasive fungi. It was suggested to use it locally. We take a liter of ordinary mineral water and put about 50 or 100 milligrams of amphotericin B and instill locally on alternate days for 3 weeks. That reduced the incidence of recurrence of the polypi. This is for information. All of us should work on the actual immunological change that is happening because of the virus or because of the treatment or because of our various methods, oxygen and so on. We have to definitely know the root of it. Otherwise, we are landing into a big trouble. Thank you.
Thank you, sir, for your observation. Dr. Sridhar, would you please respond to that?
Yeah, no, I think Dr. Appara makes very important points. I agree. I think early extensive debridement is the way to go because once it gets out of control, it's very difficult to stop it. I think you make very good points there. In terms of why is it surging at this? Number one, we know the mucormycosis enters the tissue through one single receptor, which is the glucose regulatory protein. We also know that certain conditions upregulate this glucose regulator protein through which mucormycosis enters the cell. One is a high glucose. So I think when we're managing COVID, you must make sure that the serum glucose is always below 250, optimally below 200, because we haven't seen a whole lot of mucormycosis in patients whose glucose levels are above below 200. We haven't seen that. The second thing is obviously you want to avoid iron overloads as much as we can because free iron is how the fungus grows on, and we have to prevent giving iron and zinc supplements. Third thing, acidosis. That's the third reason why glucose regulatory protein gets upregulated. The lower the pH, the higher the amount of glucose regulatory protein, and mucor enters through that. There's intriguing evidence that there are certain people who are looking at two important pathways. One is whether COVID actually somehow upregulates the expression of glucose regulatory protein, particularly the Delta virus. So that's something people are looking at. And the second thing is, I think one of the somebody in the audience 2 weeks ago asked me whether statins are protecting people against COVID. Use of statins, they saw fewer cases of mucor in people who are taking statins. So there is a group of investigators who started looking at it, and their preliminary evidence is showing in that direction. For some reason, people who were on statins had lower rates of mucor, and this is very preliminary, very early evidence. And it may be somehow statins is decreasing the expression of glucose regulatory protein through which, and that's how my mucor is also going down. So these are all areas that we need to look at and explore more. But until that information is available, we need to make sure that the glucose levels has to be below 200, avoid iron overload, and avoid metabolic acidosis, especially the chronic kind, at all costs. As somebody wrote that, well, they were not diabetic, they still saw it. You don't have to be diabetic to have high glucose. Firstly, infection itself raises your glucose level, and the steroid levels, 6 milligrams of dexamethasone, will raise steroid levels in many people over the age of 50 to 300. And so you don't need to be diabetic to have your glucose to be greater than 300. So non-diabetics also, 6 milligrams of dexamethasone will raise it. And if you give it beyond 10 days, you already are prone to develop it. So therefore, until we know more, we should keep glucose, serum glucose levels below 200 at all times. That's the point I want to make.
Thank you, Dr. Sridhar.
Yeah.
Thank you, Dr. Bhagyalakshmi, for allowing me to speak. Thank you.
Sir, 1 more minute, sir. Principal,
Yeah.
sir.
Yeah, go ahead.
Sir,
Go
I'm
ahead.
in touch with Amkana, sir, Nagendra Mainani, Mallareddy, Dr. Mallareddy, Dr. Prasad Chalsani regarding the samples for the last 1 month, sir. They tried, Sridhar, sir, Sridhar Chilmuri, sir, they tried all means, sir, from Canada, from Philippines, from Taiwan. Unfortunately, in spite of all their efforts, things didn't work out, sir. I also sent them a list of all the Indian companies which are cleared by central government to manufacture on an emergency basis this amphotericin B, sir. But unfortunately, government of India and state governments only purchase all the 100% of their produce, sir. Due to the situation here, no private purchase is allowed, sir. No private purchase allowed. So many companies are given clearance in India on an emergency basis for the production of this amphotericin B, but all 100% produce is to be purchased by government. That is the rule, sir, as the existing rule. But I told Prasad Chalsani to try on one another point, sir. Though it is not private purchase, because Amkana is an NPO, non-profit organization, and the shipment is to AMC and KGH, which is a general hospital. You can try on that basis also. Last resort, I suggested to Prasad Chalsani, sir. That is the present position, sir. I'm in touch with them for the last 1 month.
Yeah, I contacted, we do large pharmaceutical purchases for the hospital. So that's how I called the local distributor, and he was willing to provide us with doses. So once the shipping issue is resolved, we'll work it out. We'll make sure some amphotericin B comes to KG.
Thank you, sir. Thank you, sir. Thank you.
So let us see the chart box. So someone has mentioned how many of 160 cases admitted in KGH are non-diabetic and any pediatric cases. So far, we have not encountered pediatric cases with mucormycosis. Dr. Shekhar, I think he can as well address that.
Yeah, Shekhar, are there any non-diabetics who are admitted in our wards?
Sir, our medicine PGs were saying, sir, some 97 or 96% were diabetics, sir. Just there's a gross estimate, sir, total, not the detailed studies, sir. They are not the detailed statistics, sir, but they are saying they hardly came across a non-diabetic case. They said some 97%, sometime back, sir, our PG is working in mucormycosis wards. They were saying like that, sir.
True,
97.
that's true.
Vijay
Almost
Shekhar, please clarify. Please
yes,
clarify.
yes, sir. There are a few non-diabetic also, sir. Exact percentage I do not know, but 4 or 5 patients. So far
Yes.
we have attended more than 120 patients. So 3 or 4 patients are non-diabetics actually.
Yes, sir. That's what our PGs were also saying. Yes, sir.
And also, interestingly, 2 cases have come non-COVID without a history of COVID, sir. I do not know
Yes.
the relevance. 2 non-COVID mucars also have come in this last one.
Yeah.
Yes.
Someone, Dr. Satyashish has mentioned, we came across 3 cases who are less than 30 and non-diabetic. Total 3 cases of non-diabetic cases we have encountered with mucormycosis.
So on the state committee, right now I think the total number in Andhra is close to 3,000. But when the number was around 1,400, the report that we had received was about 800 were diabetics. So there were a significant number, about 300, 400 even back then were not diabetics. Only about 600 were actually on some kind of oxygen. So that was at 1,400. And also, even back then, there were about 150 patients with mucor who did not have any exposure to COVID or any history of COVID. So we still don't know, the first point. Second point is to Dr. Sridhar's thing. Now I think there's emerging data from UK where the Delta strain is a predominant strain, that it is certainly, along with being more transmissible, the rate of hospitalization also seems to be much higher than the Alpha strain in UK. And I think we're beginning to see that in US as well. In US too, I think the Delta strain is going to be predominant strain if it is not already. And the interesting thing is in UK, where there's significant amount of patients with Delta strain right now, they are not reporting, mucor.
I want to correct that the UK surprisingly started showing mucor. So they got their first cluster of mucor in UK with the Delta.
Oh, is that right?
strain.
Okay, I didn't.
Yeah, they're beginning to report. That's why I was getting worried that the Delta strain somehow raises your risk for developing mucor. Both UK and another, I think Switzerland or somewhere, they reported their first cluster, or the Holland, I think, reported the first cluster of mucormycosis, and they believe all of them had the Delta variant. So probably won't see the numbers we saw in India because they're much more careful about the steroids and how they maintain sterility. But I think there is something with this strain that increases the one's chances of getting mucor. So they are seeing it there too.
So has UK actually published the data on mucor?
So I think they are now, as news reports that came, that they had seen 5 cases, which they didn't see in the previous wave. So they'd say they're probably sicker, and that's why they may begin to see more mucor now. So yeah, so it's not in a peer-reviewed publication, but there were news reports from there, and a friend of mine also told me that they saw their first. We haven't seen the worst of this virus. It will continue to evolve and continue to teach us new lessons.
I think with that, Dr. Bhagyalakshmi, you can end the session.
Okay, sir. So thank you very much, Dr. Sudhakar, for giving me this opportunity. I profusely thank Dr. Sridhar and Buddharaju and Dr. M. Apparagaru for sharing their expertise. And I sincerely thank Dr. Upendra for his very excellent lecture. So with this concluding remarks by Dr. Sudhakar.
Yeah, yeah. Thank you very much, Upendra, for a wonderful presentation. And Sridhar and Buddharaju for chipping in and making significant contributions, particularly with regards to where we have to go and how we have to handle this particular situation. And next week, we are going to have the 75th episode, which is a very important milestone in this series. And I'm very glad to announce that two highly rated and excellent speakers will be coming up from CMC Vellore, a neurosurgeon and an ENT surgeon couple, dealing with their experience. They have the first-hand experience of handling this particular situation, and they'll be talking. Dr. Vedantam Rajashekar and Dr. Rupa Rajashekar, both of them will be talking about the same mucormycosis, the ENT, and the neurosurgical perspective. And I think that will be a very interesting session. I request all of you to log in again next week at the same time. And until then,